Hello, I'm Xin Cindy Sun, Scientific Editor at the Journal of Experimental Medicine. As part of the Keystone Symposia virtual meeting on advances in cancer immunotherapy, today I have the great pleasure of interviewing Dr. Georgina Long. Dr. Long is the co-medical director of the Melanoma Institute Australia, where she is chair of melanoma medical oncology and translational research. In addition, she is a medical oncologist and clinician-researcher at the University of Sydney's Royal North Shore Hospital. She leads an extensive clinical trials team and laboratory with a focus on targeted therapies and immuno-oncology in melanoma. Thank you for joining us today, Dr. Long.
Thank you, Cindy. It's great to be here.
First of all, I would like to ask you, as a physician scientist studying melanoma, what are the most exciting and promising new advances in melanoma treatments?
I have to give a little bit of history. It's been a very exciting time for melanoma and melanoma research, particularly at the clinical front and in the laboratory. 10 years ago, patients who had advanced melanoma, that's stage 4 melanoma or melanoma that spread around the body, for example, to the lung, the brain, the liver, would universally, it was a fatal disease and on average would survive 9 to 12 months. And we didn't have much treatment for them at all. So it's been a very exciting decade, particularly with the immunotherapies, which has more than tripled the survival of patients. Now patients are surviving at 5 years, 50% of patients are surviving, where it used to be only 5%. So I think the most exciting advance really was the checkpoint inhibitors. And we're now at a phase where we're building on that and trying to help the next set of patients who aren't being helped by checkpoint inhibitors. The most exciting thing now, though, right now, is how do we use these treatments in a better way? So we have two major treatments in melanoma, the immunotherapies, which this symposia is about, but also targeted therapies. How can we use those in a better way for patients? And one thing that's particularly exciting is the neoadjuvant application of these drug therapies. So once we discovered that these drugs work beautifully in advanced or metastatic or stage 4 melanoma, we did trials to show that they worked in early melanoma, high-risk early melanoma, that's stage 3 melanoma. And now we're doing trials where we use these drugs before surgery. So in high-risk early melanoma, usually it used to just be managed by surgery alone, no drug therapy. Now we're giving patients drugs first, then we do the surgery. So it does two things. One, we're finding that patients are doing a lot better. They're surviving better, not recurring from their melanoma. Number two, it gives us excellent material to work in the laboratory to understand resistance and great response. But our focus now is on resistance because we get the tissue when the surgeons remove the melanoma after 6 weeks of drug therapy. So that's called neoadjuvant treatment, and it is fantastic for patients and fantastic for translational research.
Thank you for that. So melanoma is like the post child for immunotherapy in cancer. Now we've heard about the exciting new advances, but what are the current hurdles a cancer like melanoma is facing?
That's a really important question. And melanoma certainly is the poster child for immunotherapy. We have been using immune mechanisms to fight this cancer for decades, but really saw the greatest success with the checkpoint inhibitors. Our biggest hurdle are the patients who do not respond to checkpoint inhibitors, but particularly patients who do not respond to combination checkpoint inhibition. So in melanoma, what we call our currently best treatment, our Rolls-Royce treatment, would be combination of anti-PD-1 checkpoint inhibition and anti-CTLA-4. However, about 30% of patients do not respond at all. And we've coined the term super progressors because they have primary resistance to this drug combination. So the biggest hurdle we're facing is this primary resistance in combination checkpoint inhibition. And that's our major hurdle. I think the great thing is, though, that what we can learn in melanoma, particularly with resistance, we can apply to other cancers that are immunogenic. So that's our greatest hurdle. What we're doing about it, as I said before, the neoadjuvant platform is one of the best ways to actually look at this problem, because we get a large amount of tissue and apply these drugs before surgery. And we're finding some interesting results, if I can touch on that, if I may. First of all, the super progressors tend not to have much T cells at baseline. Now, that's not that novel now, that's well known. But we're looking at the geography of the tumor and how the immune cells within the tumor are interacting with the cancer cells. And we're seeing that distance makes a huge difference. So having a lot of immune cells that are very close proximity and almost in contact with the cancer cells bodes well for patients. So you can still have some, for example, T cells, important subset of cells for dealing with the tumor and immune mediated cell kill. But if they're not in very close proximity, and we've actually measured how close, and don't have certain receptors, they won't be very active. Mm-hmm. So when we looked at primary resistance, we found the vast majority of patients with primary resistance don't have T cells. However, there are a subset of patients that do have T cells in their tumors, but we just can't seem to activate them. It's only a small subset. But the good news is that they do have other hooks in the immune system that we could use. So they do express other checkpoints that are the phenotype of a more exhausted T cell. So a TIM3, TIGIT, LAG3. So that's exciting, although it's not capturing the whole problem. It's only capturing a small subset of primary resistance. And now we're turning to the ones where we have no T cells whatsoever and what is going on there. It's interesting, we were looking at another subset of our patients. We have a lot of patients. We treat about 2,000 melanoma patients per annum at Melanoma Institute Australia. And we found that molecularly, we do have things we can use. So if they're not responding to the the CTLA-4 and PD-1 combination, and there are no T cells, interestingly, over half of them had either an NRAS or a BRAF mutation that we could use with targeted therapy. But regrettably, targeted therapy doesn't give us the same durable control of the cancer. So that's where the hurdles are at the moment in melanoma, and that's primary resistance to combination immunotherapy, super progressors.
How are you and your colleagues in the field tackling these hurdles or overcoming them?
So that's a great question, Cindy, and I touched on some of the things we're doing, particularly that super progressor population and looking at their T cells and how we can activate them and why they're not there. But the other thing we're doing is we're looking at what we call biologically interesting patients. So rather than looking at large groups of patients and tissue from large groups of patients, we're saying, okay, of this data set of 100 or 200 patients, who are the really biologically interesting ones? So for example, patients who have a really high interferon gamma signature. So we know that checkpoint inhibitors work well when we've got a lot of T cells in the tumor, when we've got an interferon gamma signature, and that goes with a high tumor mutation burden as well. We know that, that's well documented. So what we're doing is we're saying, okay, in this bunch of patients who have a high interferon gamma and high tumor mutation burden, where we expect them to respond and they're not. So they're what we call biologically interesting or discordant, biologically discordant. Why is that happening? Because we think the way to fast track information is to really delve into individual patients that show really interesting biologically characteristics. The other group that we looked at are those who had a high interferon gamma and low tumor mutation burden, and those that did not respond, why did they not respond? And vice versa, high tumor mutation burden, low interferon gamma signature, why did they not respond? And we found some interesting things. This is not yet published, it's under review at the moment in a journal, and we found some interesting reasons to explain why these biologically interesting or discordant, distinct patients were not responding when we predicted them to. On the other hand, we then took patients who had a really low interferon gamma and low tumor mutation burden, who we predicted would not respond, and they responded. And we thought, well, what's happening there? Why are those patients responding? And we found, again, some interesting insights into why these patients who did not have these features were responding to checkpoint inhibitors. So I think the future in tackling these hurdles, as I said before, it's about getting the tissue and exploring it, but even more importantly is looking at the individual patient story. Because one thing we do know is that resistance to checkpoint inhibitors is very, very heterogeneous. So we're now delving into individual patients or those biologically discordant, interesting patients. And if we can understand them, maybe we can understand more about others.
Yeah, I look forward to reading this manuscript soon. So now we've talked about what we've learned about melanoma immunotherapies. How might these lessons we learned be translated to treating other cancers, and which type of cancers?
That's another great question, Cindy. And as I said before, melanoma is the poster child, and as you've alluded to, poster child for immunotherapy. And there are some other cancers that are immunogenic, like melanoma. So, for example, renal cell carcinoma, a subset of patients with non-small cell lung cancer can do very well with immunotherapy, bladder cancer, head and neck cancer. So I think because we've been using checkpoint inhibitors in melanoma for so long, we've accumulated a lot of tissue and data, patient data, and that can be leveraged for the other cancers that are similarly immunogenic. I was going to mention something that we found very interesting that's going actually to be presented at one of the clinical conferences, the European Society of Medical Oncology, is we're also finding activity of checkpoint inhibitors in other cancers that we hadn't really expected to, and that will be presented at ESMO. For example, Kaposi sarcoma, there's going to be data on that tumor. Also BCC, so basal cell carcinoma. That tumor we usually treat with hedgehog inhibitors, and there is some activity of anti-PD-1 therapies after hedgehog inhibitors. So I think, and that's mainly because of the melanoma experience, that we're using this in the other skin cancers, but also those other immunogenic cancers. So I think the main point is the resistance mechanisms, I believe, are going to be similar across all cancers. It's going to be a little different proportion of the resistance mechanisms in each of the cancers, but I think at basic level, they will be quite similar. So what we find in melanoma, I believe, will be applicable to those other cancers as well.
Yeah, so along the same line, then how are the lessons we learned in treating other cancers guide advances in melanoma immunotherapy?
That's a great question. And I think I can illustrate that best. So the question is, what about other cancers? Yes, melanoma is the poster child. We've been using it for decades, immune therapy. We're the first to be using checkpoint inhibitors successfully. What can we learn from other cancers? And I think the best example I can give is probably the renal cell carcinoma group. And because what they are now doing is that they are combining the immune checkpoint inhibitors with targeted therapies, which we're all doing. We're doing that with BRAF inhibitors and various other targeted therapies. But they're finding success with VEGF inhibition, or these tyrosine kinase inhibitors that inhibit multiple tyrosine kinase but have a lot of VEGF inhibition. So from the renal cell carcinoma group, we're learning a little bit about their combinations of targeted therapy with immunotherapy to try and improve patient outcomes. And so we've recently been looking at a VEGF inhibitor with PD-1 combination. Again, will be presented at ESMO, and very exciting results in those super progressors. So these super progressor patients, it's heartbreaking. It's they come in full of hope. They know that there's a high chance in melanoma of doing well on checkpoint inhibitors, but 12 weeks later, their scans are showing everything's growing. They're getting sicker. They're feeling really sick. They're often young patients, and within about 9 months to 12 months, they die. And so we're now starting to see a few exciting results with a VEGF type of inhibition. But that will be presented at ESMO. And you have to remember, these super progressors, nothing works. Absolutely nothing has worked. Intralesional therapies do not work in this group. We're talking these really very aggressive cancers. And so we're seeing a signal, which will be exciting and presented at ESMO. Durability will be the important. Durability of response and good clinical outcome will be the big question there. But at least we're starting to see some signal for these super progressors.
Yeah, it's heartbreaking to hear about this not as promising outcome from this patient. And I guess now, especially when we are all facing the health pandemic, I would also maybe want to ask some more general questions. So are patients with melanoma or cancer in general at higher risk from COVID-19? How are we adapting cancer care to this pandemic scenario?
That's an excellent, excellent question. In Australia and New Zealand, we've been quite lucky in that we got the wave of COVID a little behind the rest of the world, which means we were able to put in place a lot of public health measures that prevented the spread. So we haven't had the same overwhelming cases of COVID that other parts of the world have had. So in practice, it hasn't impacted our clinical practice as much, although it has. It's been very exhausting in that you've got to put in new measures and everyone has to wear masks, or you have to do telehealth, et cetera, et cetera. So it takes more energy in the era of a pandemic, a lot more energy to give good clinical care, because you always have to find roundabout ways of managing patients without coming in contact. So it's a lot of energy, a lot of emotional energy, a lot of mental energy to do the planning, and constantly changing, and the waves coming and the second waves coming, etc. So that's the first point. It's exhausting for everybody concerned. But in terms of cancer specifically and COVID-19, we do know from large studies, and again, we have to be careful because in this era of the pandemic, we're gathering data as quickly as we can, and it may not be done as carefully as we would have in eras where we don't have a pandemic, because we're trying hard to get the information out there for the world. We're all pulling together with this pandemic, and we all want to share the information to try and hopefully impact positively across the world. That's all we want to do, which is wonderful to see. So we have to be careful with the data, but the data suggests two things. I think it's really important that we understand this. And I'm going to separate cancer patients and then immune checkpoint inhibitor treated patients, because it's a different question for them. First of all, cancer patients do not seem to be more prone to contracting the virus. We know that. They're not going to get it more often. If they follow the good public health measures, they're at risk of contracting it just as everyone else. The second point, though, is if they contract COVID in general, cancer patients do not do as well. They have a higher mortality rate. Now, and it's around that 8 to 10 to 15%, depends on the study. But it's very difficult to really tease that out, because there are some cancer patients who have no active cancer, but they're cancer patients. That's not the group we're talking about. They're just like everybody else in the community. We're talking about people with active cancer. So advanced cancer, whether it be lung cancer, breast cancer, melanoma, et cetera. But if they've got advanced active cancer that's growing, that's the population that, should they get COVID, they will not do so well. They don't have the same reserve. Their lungs don't have the same reserve. Their body, their organs don't have the same reserve. So they do have a higher mortality. However, if they're doing well and patients don't have active cancer, or they've had checkpoint inhibition a few years ago and they're in complete response and they're doing very well, they are not at higher risk of dying from COVID-19. We looked at that in a study of 110 patients. We actually presented it at AACR just recently, and we're now preparing the publication. We've very, very careful study of 110 patients who were on checkpoint inhibitors, actively on checkpoint inhibitors, who contracted COVID, and how they did. Now, they had a mortality rate of just under 8%. However, the ones that died from COVID, only 7 out of the 110 actually died from COVID. the ones that died from COVID had very, very advanced melanoma or another cancer and were not doing terribly well from their cancer. So they didn't have as much reserve. So we don't think that anti-PD-1 per se in itself means that you have a worse immune response to the COVID and therefore get worse lung findings, worse pneumonitis, and therefore more likely to die. We don't think that that is the case. We're not seeing that signal, if that makes sense. It's a very different question to just having advanced cancer. You're going to be more likely to succumb to most things in this world, any virus, just because you're quite debilitated. But just being on PD-1 alone per se does not, we think from the data we have, does not seem to increase your risk of a bad lung response to COVID-19 and therefore make you at high risk of death.
Yeah, thank you for the clarification. Hopefully our scientists will learn more and help this patient. And I guess last, as a scientific editor working at a scientific journal, I would like to learn how was your experience being with scientific publishing and peer review during this health pandemic? And is there anything that you would like scientific journals to improve?
Another great question, Cindy. I have to say, luckily, most of the publication world is online. So it's adapted beautifully to the pandemic in that you can seamlessly get on with publication and interact with people. I haven't felt much of a change here in Australia, I must say. But one thing, and I alluded to it before, is the difficulty in trying to sort out what is quality research and what is a little too quickly put together, and the danger is the conclusions we come to. So if something's done very quickly, even our study, we've done it very carefully, very quickly, but we're very aware that we've got to be very careful that we don't come to conclusions that are not correct in an effort to get things out quickly in terms of the pandemic. I found the journals to be very responsive in this pandemic, keen to publish work particularly related to the COVID pandemic, still keen to publish work they would normally publish without the pandemic. And because it's been like a duck to water in terms of the virtual platforms, I don't feel it's been terribly effective. We just have to maintain scientific, high quality scientific research in terms of publication during this pandemic.
Yeah, thank you for this very positive comment. I guess as scientific editors, really during this pandemic, both us and our journal, we are trying really hard to work with the authors, the reviewers to adapt to this challenging time and allowing the authors to have more time to get quality data published, but also recognize what may not be done for this challenging time because of the lab shutdown. So we are, trying to help them to determine which are the data that should be done to improve the study, and which are the experiment that maybe we can just add them into discussion to show in the future these things can be done to further explore. So yeah, we have been doing a lot of things.
That's a really good point, actually, Cindy, and that is the impact, and I didn't mention that, but the impact the pandemic has had on laboratory ability to go and do experiments and confirm findings or extend findings. You're right, that's been a very difficult thing. And I've also found that the journals are being a lot more careful about the reviewers' comments and getting more involved in terms of saying, no, that reviewer comment will let slip. That one, yes, we do need a bit more data. If you have it, if you've already got the genomics, if it's already on a database, if it's easy. So you're right, but it's a very positive thing. I think most of the journals have done exactly what you've said, and it's been very cognizant and helpful in knowing that we can't get into the lab to do the same experiments we used to be able to do. But we still have to be careful and review the situation if and when, which I'm very hopeful, we get a vaccine and we can all get together again, and just be sure that the quality of the work that we've produced during this period doesn't need a little more refining or a little more work, and collecting data on that as well would be important.
Do you have some take-home message you want to share with our audience before we end our conversation?
Oh, that's a great question. My one take-home message is for people who are doing translational work, both the clinicians and the scientists, get together, keep talking. We can't do the work without clinicians really giving us good input. And same with the scientists. The clinicians can't move forward and find great new promising ways to manage patients without our scientists. So my take-home message is keep working together. That's our only way that we can move forward more efficiently and more quickly.
Thank you so much again for your time and the insight. It was a great pleasure talking to you today. Last but not least, I would like to thank our audience for listening to this one-on-one Q&A session. You can learn more about the latest advances in cancer immunotherapy at the eSymposia virtual meeting on August 17th to 19th, 2020. I look forward to discovering more new insights at this landmark virtual event, featuring more global leaders and rising stars in the field. Thank you.
Thank you, Cindy.